So let me first ask, are there any questions from yesterday? No? I had a question. So do you have an answer? I had this question about how much time would it take for allele A1, which is present in a frequency 0.01, to reach a value 0.99. Okay, so I'm sure you'll try it later, but I just, those who have done, I'll just give you the answer. Those who haven't, please try it. So yesterday we discussed this in the tutorial, that you should get something like this. Okay, this you can show from the discrete time equation I had written, for assuming S is very small. Okay, and I think S I gave 0.1, and if I ask initial value 0.01 to become 0.99, this basically you can integrate, right? That's why I asked you to make this approximation. And if I did my maths correctly, I got something like 1,000 generations. So see if you agree. If you disagree, that's fine. We can discuss it. Okay. There are a couple of remarks I wanted to make first. So what did we do yesterday? So we basically considered a one locus problem with 2 alleles, and only force was selection. And we worked with diploids. Okay? And the thing was that the equations are nonlinear. There are several equilibria, and you have to do stability analysis to figure out under what conditions which is the stable equilibrium. But you can just think of it as a mathematical problem. It doesn't have to be 2 alleles. It could be k alleles or a alleles. Then there are several equilibria, how many are stable, etc. The question is very simple to state, but we don't have any such general statements available. What is known is just the number of bounds, the bound on the number of stable equilibria. Okay, so I thought, so this is something which is studied by a lot of people, yet there is not too many things which are known. I mean, it's quite a bit known, but there is still some room, to do things. And if these are the kind of problems that interest you, I'll give you a reference that you can look up this nice book by Bulger. It's called Mathematical Theory. Selection, mutation, anti-convention. Okay. The other point I wanted to make, yesterday's selection scheme I considered is extremely simple. These were just 3 variables which you could play around with. Basically, this problem that we discussed, one locus to allele selection, that you can work out in great detail completely. Even not just the linear stability analysis, you can also do global stability analysis. There's a lot known for that. Okay? But there are other selection schemes. Fitnesses could be time dependent. They could also be frequency dependent. So I didn't discuss all that. So WIJs could be more complex, more complicated. Okay? There are the scenarios and the steady states, etc. There's quite a rich structure for that. Some of you were asking references for this stuff that we are discussing. They are available. I think there was some stuff I had given, which is on the website of the program. But I'll just tell you, I'll write it here also. So there is 2 books which you could refer to, Charlesworth and Charlesworth. And the other one is by Ewens. So these 2 guys are biologists. So the book has both maths and biology, but more biology. And this one has more maths, but some bit of biology. So most of the things that I say, you could find in one of these 2 books. Okay. And today what I want to do, I give you a small flavor of one locus problems. Today I want to do a multi-locus problem. So it's not just a locus, it's not just A1, A2 sort of thing, but it's just a sequence of length M. It could mean stretches, L stretches, one stretch, two stretch, third stretch over there, or it could mean a genetic sequence of length M. So when I say locus, today I mean it doesn't have to be disjointed things, it could be just a sequence of length M. So this is what I want to do today, and I want to also include selection and mutation models. So this is what I want to do. Multi-loci models with mutation, which we didn't discuss yesterday, and selection. So that's the aim for today. Let's see how it goes. Okay. So we discussed one locus. I want to actually start with a simple two locus model. And don't worry, I will not do 3, 4, 5, 6. I'll jump right to L. But for the two locus model, there is a small point I want to make. And there's a result which is, I think, appealing to physicists. There's a quantity which comes out that physicists we can interpret. They could understand it better. So that's why I thought I'll do that first. So, and I also want to describe what this term means, recombination. That's the first thing let's do. Two locus. No selection, no mutation. I'll introduce this thing called recombination. I'll explain what that means. Okay. So yesterday we had diploids, which we said were of this kind, which we represented the configurations like this, right? It's a bit more complicated, but stay with me. We'll get to something interesting, I think. I have two loci now. At this locus, there are two alleles, A1 or A2. And the other locus, again, I have two alleles, B1 or B2. Okay? So just like, so this was the diploid, and I said under Hardy-Weinberg equilibrium, I could work with alleles. That means I don't need to worry about this double thing. I could work with a single thing, that is the allele, that is A1 or A2. Here also, just by it, I can actually work with combinations of this kind. So A1, B1. This is not the same thing as this. Here, this first copy referred to the first index referred to the mom, second one to dad. Here, this whole thing is mom. This is the first locus of the mother and second locus of the mother's sequence. And if I add the same thing of the dad, you can check that you will get such 10 combinations. We don't need to bother about that. We'll just take the same attitude as yesterday, that we can work with the alleles. That means I'll just keep A1 and A2. That means one parent. Similarly, here also, I'll just keep one part. Okay, we can work with that, just like we did yesterday. So there's a parallel to that. So you get such 10 genotypes. Please remember the genotypes. And here we had two alleles. And here in this case, we have 4, it's called gametes. Never mind what these names are. Let's say the frequencies are these. Okay? So just if you keep this one locus problem at the back of your mind, this is not hard to follow. Okay? It's a little bit more complicated, but basically steps are quite similar. But here the difference is there's no selection, there's no mutation, no other evolutionary forces, except what is called recombinations. Let me say what that means. So recombination is the following thing. It's a way of genetic mixing. If I had one locus, I could not implement this force. With two loci, I can, in the following way. So let's say this is mom's genome and this is dad's. Recombination means you break this and swap the parts. You have to write all of them. A1, B1 with A1, B1 here. A1, B1, A2, B1. So I have not written all of them. So you'll see there are 10 of them. But this is only one parent. Alleles are 2 at each locus. Allele is simple. It's just saying one type. I just choose whatever. Let's say 0. And the number of variants of that are the alleles. So if I am type 0 and many of you are a 1, then we have 2 alleles in this room. That's all. So we one type, choose as a reference, and all its variants, if you write also, total number of these things will be alleles, number of alleles. Here we have 2 alleles, but 2 loci. I'm writing B instead of A there because I want to distinguish between saying this is locus 1 and locus 2. You can use some other notation if you like. This notation, if you like, you could have written something like still a two allele problem. Okay? So this is the recombination. You break it and swap it. That's how sexual reproduction. I mean, this is a very simple caricature. Sexual reproduction happens like this. The 2 genomes, parents, they break, and at some point, they swap the material, genetic material. So what this is doing is to model the process of genetic mixing. And that complicates things. Okay. So if I do this with certain probability R, I'll get something like this. Is this okay? Okay. Oh, that I didn't write. So why didn't you do this as an exercise? The genotypes would be 10 if I write A1, B1, A1, B1, A1, B1, A1, B2, A1, B1, A2, B1, A1, B1, A2, B2. Then repeat the same thing for this guy, keeping track of the fact that I'm not overcounting. Yeah, it's not very large, it's just 10. You have to write it. It's 10. Okay? There are 10 genotypes. So try it and otherwise check with me. But the recombination is this. Break it and swap it. Yes? No, this is different. No, it's not blending. It's a mixing of genetic material. Everything is Mendelian. Blending is out. No, it's not saying that my phenotypic character is a blend of my parents. This is saying sexual reproduction means mixing of genetic material. Genetic material mixes. That's a sexual reproduction. That doesn't mean that my color would be the same as an average of that of parents. Okay? But this is just mixing the 2 genes, the 2 genetic materials. Okay? So this would give me this new type of gametes. And just like for Hardy-Weinberg equilibrium yesterday, I want to write down equations for these 4 things, C1, C2, C3, C4. Okay, so let's try this. Okay, so C1 prime is equal to C1, nothing happened. With probability R only, recombination occurs. And do you agree with this? So C1 is the frequency of A1, B1. I'll get a contribution to that. So I want this guy. It's again the sort of thing we did yesterday for Hardy-Weinberg. I want a contribution to A1, B1 frequency that would come from this, and their frequencies are C2 and C3. Is this okay? Yeah. But what about this one? Okay, why don't you tell me this? If I do break and swap, would it change anything? No? No. That's right. How about this guy? break and swap. Changes? Yes, very good. So this, when I do, it will produce A1, B2 and A2, B1. The frequencies are C1, C4. But that's the one which is reducing the frequency of C1. Okay? Okay, it took me a while to write it, but I'm sure you can do it. Okay? And this kind of things you can write again as a homework exercise, you can do it. And let me define a quantity. Yeah? Small r is the probability with which recombination occurs. The swapping and breaking and swapping, it happens with a certain probability r between 0 and 1. This is that of probabilities in discrete time. Yes, yeah. R, yes. Okay. And I want to define this quantity. D. It has a special name. It's called linkage disequilibrium. LD, and it was introduced in 1960. I don't know what the meaning, why did they call this way, but okay, that's the name. Okay? If you use this, then you repeat this exercise for C2, C3, C4, you will basically get equations like this. How about you tell me this? What can you say about this quantity, given all the information? Same? It's not that easier to actually expand things and stuff. Okay? So, I'm sure you can do this, and you will see this. So, the LD in the next generation is 1 minus R of that in the previous generation. Right now, it's like, okay, so what? But just, wait for a second. So, this, of course, you can solve. The solution is just this. So why am I going into all this detail and making things in life complicated? Because this object D is something that physicists recognize. So let's see. Minus D. So I'm just writing the definition here. And let me write C4 because these the total is 1. Okay, that's fine. However, note the following. Frequency of A1 is equal to the frequency does this make sense? Frequency of A1 is, I don't care what's happening the other one, right? Second locus could have either B1 or B2. So frequency of A1, the first locus has allele A1, is irrespective of whether second locus is B1 or B2. But that's equal to C2. Can you tell me the frequency of B1? C2, C1 plus C3. And if you use this, you'll see that this is equal to this expansion that I did, for D minus D is equal to this. And what does that mean? So the point is, minus D or D up to some minus signs and all, this is equal to frequency of A1 B1 minus does this look like something you've seen in your physics classes? Correlation function, right? So it's saying the frequency in general. So this I did for 2 locus. But if I in general have a long sequence, the frequency of that configuration minus the frequency of each loci. It's like the density of a configuration, right? Probability or density of a configuration 0, 0, 1, etc. minus the density at that point times the density, etc. Right? It's of that kind of object. So this linkage disequilibrium, as a physicist, we recognize as a correlation function. This is an important object, and some effort there is expended in calculating it. But more interestingly, more importantly, if R is pretty large, linkage disequilibrium, LD, decays very fast, which means I can ignore D. Correlations are approximately, it's a good approximation, when R is pretty large. Okay? And that makes intuitive sense. So I'm saying, with probability 1, you break and swap. That means you're mixing things. There won't be any correlations, right? So when I have to do a multi-loci problem, not just 2 locus, multi-loci problem, and if I know that recombination rate is pretty high, one approximation people use is to write the joint distribution as approximately as a product over the 1 locus answers. And 1 locus answers, we have done to death. We know a lot about it. So the point of this exercise was just to point out to you that if it's a very, not promiscuous, but what I mean is it's a quite recombining population, then this approximation is a pretty good approximation. It's used in the literature quite a lot. Okay? So that's a sort of little statement connection I wanted to make here. And I mean, not just deterministic models, stochastic models, which are harder. Again, you make this make this approximation that the distribution of the product of the full configuration is product over each locus. At single locus problems, we know. We have the answers to that. Sorry? Usually not. It's actually more space dependent. So if I'm looking at this locus, R may be different. And if I'm looking at the locus, it may be different. Over very large time scales, maybe it does, but I'm not aware. Okay. So that's what I wanted to say about this little word called recombination. And there are a lot of problems here which people are interested in, but they are very, they are not easy problems. They're quite challenging and interesting. Okay, so I'll erase this bit, and I want to now do something simpler. I want to now consider L locus problem, LSI, mutation and selection. Okay. And let's have this parameter R for a second. And what I said was R is large, like it is close to 1 or so. Then you can basically appeal to 1 locus problems. But what if R is small? Then this linkage disequilibrium, it will not decay. It will remain for a very, very long time. So let me just, so this was one limiting case. Let me go to the other limit in which R is exactly 0. That means the population is asexual. So these are two sort of limiting cases that I'm thinking about. Okay, so that's what I want to discuss now. Other thing is, so this is one assumption. I'm going to make our life a little bit simpler by considering not diploids, but haploids. That means we don't care about writing things like A1, 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 A2. We just think directly in terms of A1s and A2s. There's a technical reason why I'm going to haploids. There's of course biological reasons. Some plants, fungi and all, they are haploids. Our germ cells are haploids. But there's also a technical mathematical reason, which I'll explain. I can refer you to these excellent articles that are available on arXiv. So now I want to write down an L-locus problem. The views. I'm sorry? Oh, this one is Krug. He is my postdoc. He was my ex-postdoc advisor. I got started in this business with him. Okay? Yeah. So one locus. We said if it's a haploid, I'll just say I have just one locus. It could have A1 or A2. Two locus, I said, could be A1 here, B1 here. Oh, bigger, sorry, okay. L loci, of course I can write like A1, B1 and all, but I'll run out of the English alphabet. So it's better to write it as a binary sequence. So I'll represent a sequence or an L-locus, L-loci sequence as a binary sequence. So we will not write A1s and B1s, we will just think in terms of binary sequences. Is this picture okay? Instead of A1, A2, I'm just calling it 0, 1. There's no big funda here. Okay? So how many number of sequences would I get? 12. And the object of interest, as before, is the frequency of these sequences. So today I'll be using letter X. How does that evolve? So you want to again write down the same kind of discrete time, continuous time, etc. type of equation. So it's the frequency of type sigma of sequence sigma at time t. How does it evolve? What happens to it at large times? How does it change in time? So these are the same kind of questions that we're interested in today also. So let me first write down the equations. So let me just set up this bit of maths, and then we will take some specific examples. Super selection. I have this fitness. We will worry about that in another after 10 minutes. W of sigma. What I wrote as W11, W12, W22 yesterday. It's a W of sigma. I have 2 to the power L such numbers to assign. Okay. And now I also have one more force, mutation. Mutation again I'll talk about a bit later, but it's simply that a sequence can change into another with some probability matrix. Sigma prime can become sigma with some probability m. What is it, etc. I'll come in a few minutes. So let's write down the equation for this object. So that's, I have sigma prime at time t. It has a fitness W. So W has an interpretation of average number of offspring produced per generation. You can't see that right now. Tomorrow, Thursday, when I come to stochastic models, it would come out. So this is a fraction of type sigma prime. It produces this many offspring. And if I want a contribution to type sigma, mutation occurs. And we want the total contribution. So this summation is sigma prime. It's very similar to what we wrote yesterday, except we didn't have this term with m. Okay? I have a proportionality constant here because this is 1, right, at all times. So if I sum over both sides, what do I have? This is 1. Okay? So there's some constant in the front. If I do a sum over sigma, what would I get? So it's the probability that sequence sigma primes becomes sigma. So if I sum over sigma, is it? No, not conservation probability. If I start with some sequence, any sequence, I'll go to some sequence or the other, right? That's what probably you meant. Okay. So this is true. Right? This makes sense? I have some sequence and I mutate. It makes a change. 0 becomes 1, 1 becomes 0. I go to some sequence or the other. I find summing of all sequences. This is 1. And if you use that, then you can figure out C is nothing but inverse of this. Right? So in reverse, we have the equation. Object has a meaning. Can you see? It's the fitness of the type sigma prime times the fraction of type sigma prime added over all sigma prime. What is it? Huh? Louder? No, not relative, it's average fitness. I do not by W bar. Okay? So this is our equation, and we want to stare at this equation today, at least for some time. So as like yesterday, we have a nonlinear equation. Because I want to consider steady state, I have a nonlinear equation, and there are these 2 to the power L such sequences. Yeah. It's difficult. But this equation for haploids, it's not really, it's a nonlinear equation, but there's a very nice transformation which makes it linear. And I want to tell you that. Let's try this. So let's do this change of variables. plug it in this equation and tell me what you get. Shall we do it? If you plug it in here, so x sigma t is at sigma t plus 1 divided at product. So, this nonlinearity is gone. I have a linear equation in Z. Okay? Now, you have been seeing these kind of equations, although in continuous time. This is not really difficult, it's not very different from what you have been seeing the last couple of days. I have a matrix, we have this vector at time t plus 1, z1, z2, z so on, for all L of them. And there's this matrix of this type, and then there's this vector at time t. And you could try the usual standard methods to analyze this problem in principle. Only thing that you should remember is summation over z over sigma is not 1. This was one point. And suppose I have solved for z sigma. How do I get x back? If I sum over sigma on both sides, is this okay with everyone? No? Oh, okay. Okay. Sorry. Okay, so this much is okay? Yes? Okay, just okay. So all I've done is to plug in this transformation, which is z is equal to x times product over w bar at t prime, which goes from 0 to t minus 1. And if I plug it in, denominator drops out. That much is fine. And I'm just saying, how do you reverse this transformation? So here we have x here, we have x here. So if I sum over both sides, I find that x sigma t is equal to z sigma t divided by z sigma t summed over sigma primes. I'll just write it back. Better? Okay? And also, yeah, I should also mention that these elements of these matrices, they are non-negative, mostly positive, and by Perron-Frobenius theorem, we know there will be a unique stable equilibrium. So for diploids, I had to worry, there are several equilibria, and which one are stable. For haploids, because of this transformation, I know there's a unique steady state. I don't have to worry about that. Okay? For diploids, that's why the problem is harder and interesting. We don't know a transformation analogous to this one for the diploid case. Okay? So for diploids, we don't have many general statements. For loci, if you give me, we don't really know much. We can't really do much. Yeah, and you have to basically do it numerically. But there's really nice, some interesting rich structure in those models. Okay. And to just finish up this bit, I'll try to write bigger. Because this is something you've been seeing for the last couple of days. So this is a linear equation, and you've been seeing it in continuous time. Just like Martin did in today morning, he made a discrete space-time equation into continuous space-time equation. We want to keep space discrete. In all these models, space is discrete. Time you can make continuous. And I'll just tell you what you have to do, steps I just mentioned, and you should work it out yourselves. So you just do this change of variables. You define this W in terms of this new F. The delta T is small. So you take all this, plug it in over there, and do the usual Taylor series expansion. If you do that, you will wind up with this continuous time equation. There's no unique continuous time equation in these models. I'm writing one of them because it's easier to handle analytically. So the equation you will get is this. Once again, you can get this is again the offending with the problematic part. F bar is equal to this. Okay, again is the analog of that transformation to get rid of this. That's the kind of equations that you've been seeing this morning and yesterday. These are continuous time equations, and again, these are linear ones, once you got rid of this guy. Okay? Okay, so fine. So we're going to stick with, so there is some decent chance that we can make some progress with haploids, multi-loci problems. So let's do that and see what we get. Okay, but before that, I have to specify what these W's and M's are. So let's talk about that. Sorry? Argument for what? F bar is the analog of this W bar. Oh, this is the, argument means tau, yeah. It's a time. It's a rescale time. Yeah. The Z's one? No, because it's W. No, because the W is there. If W is not there, and I'm summing over sigmas, Z is not steady state. Z need not be 1. Summation of Z is not 1. Z is not normalized. It actually does, yes. Yes, yeah. Yes, it goes exponentially, and yes, that's true. Yeah. But if you look in continuous time, again, it becomes 0, and the other ones are smaller. Are negative, yeah. Okay, so let's talk about this simple part, which is the mutations. Mutations, again, I think you know roughly what it means. It just means 0 becomes 1 with a certain probability mu. And for simplicity, I'll say the other one is also mu. In general, they can be different. This is for per base or per locus, as we've been saying. And these typical values of mu are pretty small. I mean, there's like 10 to the power minus 4, which is the highest for a certain virus is called RNA virus. And for humans, it's like, or Drosophila, etc., it's like 10 to the minus 9, 10 to the minus 11. It's a very, very tiny number. Okay? So if I say 0 becomes 1, 1 2 becomes 0, this is the class of mutations which are called point mutations. That is, at each point, you ask for it. There's a change. But there could be big chunks. A mutation could be such that the big chunk of sequence is removed, or a big chunk can be added. There are those mutations, but the probability is very low. So I'm not just going to consider them at all. Okay, so we'll just concern ourselves with these ones. So I want to bring in this concept of what is called having space. So let me explain. So when we have L equal to 1, we had two sequences, 0 and 1. And I have L equal to 2. So L, remember what I said? A sequence is denoted by a string of L letters, and each sigma is either 0 or 1. Think of spins, Ising spins, right? So then, I'm going to draw this. I have 4 sequences, I'm putting them in a square. Why am I putting it? I'll explain. If I have 3, I'm going to put them in a cube. And if you want to do for L equal to 4, please do it if you want. It's very complicated. Okay, so in L, for L in general, for L, I have a line, I have a square, I have a cube. In general, it's called a hypercube. I have an L-dimensional hypercube. And sometimes also referred to as Hamming space or Hamming, I don't know, Hamming cube. Okay, this kind of objects are said to be in what is called a Hamming space. So what really concerns us is the following. So I want to write down that M that I mentioned in that evolution equation. So M between two sequences is the following. Suppose I want to mutate this sequence to this one. And I assume that each mutation happens independently. 0 becomes 1, 1 becomes 0 with the same probability. What do you think is the probability of mutation of this sequence? Mu? Why? Mu into, right? Is it right? This changes, that is wrong. That's probability is 1 minus mu. Okay? So therefore, if you understand this, I can write this guy. It's mu, the number of changes. Where would the number of changes happen? The changes, I want to go from one sequence to another. Where can I mutate? Wherever those sequences are different. And wherever these number of differences between the two sequences is called Hamming distance. So I didn't say that. The Hamming distance is simply the number of differences between two sequences. Hamming distance between these two sequences is 1. This is same, this is 1. This is 0. So D, what is the Hamming distance between these two sequences? 1. How about, what is the Hamming distance? Sorry. Okay, so that's the sort of thing I'm writing down here. So that's my mutation matrix. Just saying each base, each nucleotide or each base or each locus changes independently by a certain probability mu. That's all. Okay? Okay. Now let's worry about fitnesses. fitness, first of all, I mean, we understand like sort of, colloquially that, fitness is something, if I'm fitter than you, then I might live longer, I might have lots of children, more than you, or I mean, survival chances might be better than you, something like that, right? So there is some sort of simple understanding, simple notion. And when we put it in a model, we define it more, explicitly, more clearly, what we mean in that model fitness represents. But when we measure in experiments, typically one measures some kind of proxy for a fitness. Okay, so typically it's measured as a growth rate. So let me just tell you a couple of experiments. Especially in microbes, which is sort of interesting. I find them more interesting. So you can, okay, so I should just say that. So we talked about the space in which evolution would occur. So this space, okay, I should mention that. So we have been talking about in StatMech models, one-dimensional models, two-dimensional models, models on a honeycomb layered lattice, or in 2 dimensions again, hexagonal lattice, etc. So you have some notion of space, right? The geometrical space, the space in which these spins are going to move around. The space, the analog of that here is the Hamming space. Okay, my population is going to evolve in this space. Okay, and now with these sequences, if I attach a real positive number, that whole thing is called a fitness landscape. It's analogous to energy landscape. I'll make this correspondence between fitness and energy in the third lecture a bit more formal. But today, you just take it as an analogy. Okay, so there are sort of, yeah. No. No, allele is just 0, 1. No. Just if you're talking about a color, that's more complicated, right? That's phenotype, but let's not worry about it. Let's stay in genetic space for the moment. It's much more complicated. And these models work there as well. Let me tell you that first of all. Okay? And let's not talk about phenotypes. That's a different set of problems. Allele is simply this or anybody else. That's all. Then I will say I have 2 alleles. So this color, the peach color, and any other color of the table. It's a mutant. And that's what I would say are 2 alleles. Alleles is just the reference guide and its variants. That's all. Yeah, sure. That's why I said in the beginning that if you have A alleles, K alleles, people consider that. Those are harder problems. And there's a rich structure there. No, it's nothing to do with mutations. It's fine. There's no problem with that. In fact, you should generalize this. Yeah, good that you asked. You can give an exercise. Generalize this. If I have A alleles, it's easily generalizable. So the things won't change, qualitative behavior won't change. Okay, you could do that, it's not a problem. Okay. Yeah, so for fitness landscapes, you can do sort of 2 things that people do. One is to make them directly, in the sense that, so this is what the typical thing is. Okay, so I can't get into details, but again, let me give you the reference here. You can find more. So this is also a nice article by Joachim, this collaborator. You can find more details there, but I'll just sort of say a little bit about this. So you can make this fitness landscapes directly in the sense that following. So what you do is, there's a long sequence, let's say viral sequence. It is about 1,000 base pair long. If you want to find the fitness of each of them, which I'll define what I mean by fitness in an experiment, I have to generate 4 to the power 1,000 mutants, a full library of all 4 to the power 1,000 mutants. That's impossible. But what people do is, they just choose a small set. Typically, it's 10 to 20 nucleotides. 10 to 20, that's it. 20 is also too high. 10-ish is the max. Then you have to generate, not 4 to the power 10, but you generate 2 to the power 10 mutants. You say this is my reference, 0000, wild type. I've chosen the reference. And any mutant of that I generate, I can do that in a lab. I got these 1024 mutants, and I measure the fitness. So how do I measure fitness? You could do several things. So for instance, for bacteria, you could just measure the growth rate. So you take this bacterial colony, you give it less glucose, so that it's sort of starving, and there will be some guys which are tough guys, and they would have more offspring. They will determine the growth rate of the population, and the fitness might increase. You could measure that. So there is a very famous experiment going on since the late 1980s continuously by the lab of this person, Pensky. Should have a look. It's very exciting work. Okay, but sort of more pedestrian way would be, let's say, so growth rate is 1. That's a measure of fitness. Another one is what is called minimum inhibitory concentration of antibiotic. So what you do is, you take this bacteria of a certain type. It has, let's say, sequence 0100. You prepare that mutant. You add a little bit antibiotic. The guy is fine, it's still growing. Okay, next day, I'm a bad guy, I put more antibiotic. I keep doing that till it stops growing. So that is also a measure of fitness. So these are the kind of proxies that you devise to measure the fitness of a population, fitness of a sequence. Yeah. So for that, you have to do something called mutation accumulation experiments. And for that, you need to know a bit of stochastic theory. I'll come to that on Thursday. Okay, remind me if I forget. Okay, so we know these numbers well, okay? Okay, so I can measure, I can just take a small library and measure the growth rate or some proxy for fitness like this and assign these things. This one is, of course, labor consuming, but then you know precisely in that environment what is the fitness. So this fitness landscapes can be constructed. On the other hand, we know the fitness landscape is huge. 4 to the power 1000 sequences it has, but we know a tiny bit about that. Other way of doing it is indirect measurements, in which what you do is just to run an adaptation experiment. So I'll give you one example. So the one this is also a very nice paper. And if I get a chance, I'll show you some pictures. Nature or science something. This is it's a very old experiment. There are lots of them, but I've chosen to talk about one. So indirect way is the following. You take a colony, you take a mutant, you know its sequence, okay, and you know its fitness, initial one, and let it grow in whatever environmental conditions I have given. So for instance, in one experiment on a bacteriophage, it's a virus, which eats up the bacteria. So what they did in this experiment was, so it's a bacteriophage, it eats bacteria. So what you do is, plate a lawn of bacteria, okay, and then you drop a virus particle, and this virus will eat it up. And what it does is, when it eats it up, and there's some nice pictures, you can see, in concentric circles, the viral colony would increase. That area you can measure, that's a measure of growth rate or the fitness. Okay? So that kind of fitness you can measure. So for in one of the experiments of these guys, for example, as a function of time, when they measure the fitness, this is the average fitness of the colony, they find that sort of it increased when they started with certain sequence. When they started with a different sequence, but initial fitness was more or less same. It was sort of, plateaued, and they looked at it a little bit longer, then it sort of tried to decrease. That's the kind of information they have. They don't have all these 1024 numbers, but that's the kind of data they have. But then, looking at this data, you can infer qualitative features of the fitness landscape. So for example, the interpretation. So it's sort of a bit subjective, but you can interpret the results of this experiment as, so I'm going to draw a cartoon of fitness landscape. So here, I mean sigma, and this is W of sigma. This is just a cartoon. When I say sigma in quotes, what I actually have is this kind of thing. Remember that. Okay, that's very important. But as a cartoon, the interpretation of this sort of thing is that the population is sitting somewhere at the bottom and it's going up. And so its fitness is increasing. But this guy, which is a different fitness, it is sort of already at or near a peak. So something like that, they hypothesize, is the fitness landscape of this experiment. So this is the interact, it's incomplete, but it's exploring a lot more space than just the tell loci. So the point I'm making here is, you can get some information about fitness landscapes, some very short range structures, some large ones, but of course that comes at a cost. That means you don't know all about it. But the ones with short range, the short ones, you know, I mean, you know everything, but then it's just a tiny bit. So there is sort of, you have to see what exactly you want. And as I said, there's a nice review article. You should have a look at it. Okay, so this is what a simple bit I wanted to say about fitness landscapes. And, okay. Okay, let's do it, it's fine. Okay, so fine, lots and lots of experiments have been done and being done, etc. What have we learned? So one lesson that we have learned by these experiments is that the fitnesses are such that there are interactions between the loci. So let me try to explain that. So there is an object term called epistasis. It's a measure of, it says it's the fitness effect of a mutation. I'll explain that. It is dependent on the background. When we say there is a non-zero epistasis, it's a measure of interactions. So let me give you an example of what I mean. Okay, so let's take one. So suppose I give you a fitness scheme of this kind. Let's say three locus. So I have built a model of a fitness to be this. For a sequence, this is my model to start with. So if I have something like this, this 0 is becoming 1. This is the mutation. The rest of the guys is what I mean by background. So when I do this mutation, then if the fitness ratio does not change, it does not depend on whether I have 1, 1 here. When I do this mutation, if the answer, the ratio of the fitnesses, it does not depend upon whether I have a 1, 1 here or a 0, 1 here, or the other 3, 2 combinations, then I would say there's no epistasis, there's no interaction. Okay, that's the simplest model, and it's very, very commonly used because it simplifies a lot of stuff, and things are exactly solved. So, but let's just understand that. Can you please tell me what is the fitness ratio according to this fitness scheme for this case? How much is it? W1, 0, by W1, 1, right? What about this guy? Same, right? So, for this fitness scheme, there is no epistasis. Each locus is contributing independently. It's a product over each locus, right? Okay. So, when do I have epistasis? So, let me give you some examples. Suppose K is the number of ones in a sequence. Okay. Then for the first sequence, alpha is any number between 0 and infinity. How much is it? Number of ones are 2. So let's say alpha is 2, say, for simplicity. So K is 2, 4, and there are 3, 9. Right? What about the other one? How much is it? K is 1, 2. Ratio is not same. Except when, again you should try, alpha is 1, then there is no epistasis. Sorry, it's a bit doing it a little fast. But please try it yourself. If alpha is 1, there's no epistasis. I'm back to that case. If alpha is less than 1 and greater than 1, there are different answers. I mean, there are different types of epistasis people say. This is more commonly seen in experiments. There are evidence for both, but this is more common. Okay. This is not all. I just wanted to do this one last bit about the fitnesses. Let's just take the case of this 2 locus. It's easier to draw. So the kind of epistasis you have here is called, I'll just give you the names here. So I'm drawing an arrow in the direction of increasing fitness. This is L equal to 2, so I start with 0, 0. The fitness here is higher, that's why I put an arrow upstairs. Fitness here is higher, and then again higher, and here, and the side is higher. The magnitude may be different, but starting from here, I could find a path without ever being stuck. It's an interaction, but only in the numbers. So there's a certain name for it. And this model is for magnitude epistasis. But there are other ones which are sort of more, I think, interesting ones. It's called sign epistasis. Okay, so let's just have a look. So if I keep the background to be 0 and I make a change from 0 to 1, the fitness is decreasing. So I said increasing is, the arrow is in the increasing fitness direction. So in this case, fitness is decreasing. So such a mutation is deleterious. It's a bad mutation. The word is deleterious. On the other hand, if I make a mutation from 0 to 1, but the background is 1, okay? So the background is 1, and I'm making again 0 to 1 change. In this case, mutation is beneficial. So there's clearly epistasis, right? I said the answer depends upon the background. But if the sign, here it's a decreasing fitness, here it's increasing fitness, right? The sign of the, the ratio, that's changing. That's why it's called sign epistasis. It's not about the numbers, but in one case it's beneficial, and the one is deleterious. This is something which is again seen commonly in lots of experiments which are documenting these things. And finally, we have what is called reciprocal sign epistasis. I'm saying all this because people are classifying the fitness landscape, the data that's coming, according to some of these criteria. That's why I'm spending time on this. Does it look different from that one? What is the difference? It's also ulta. It's the opposite, right? This is interesting because what you see, the fitness is increasing this side from whether it comes from here or here, fitness is increasing. Similarly for this guy, whether it comes from here or here, fitness is increasing. I have 2 fitness peaks. The sort of thing I mentioned in one of the experiments. Right? So one of these is a local peak, and the other one is a global peak. Reminds you of physics literature, spin glasses, disordered systems. Let's say this is local. Such fitness landscapes, if I were to do in L dimensions, an L hypercube, these are called, there are lots and lots of peaks, they're called rugged fitness landscapes. So think of Himalayas. Something like this, in which you have just one peak, like this. So this is a single peak. It is named for it Mount Fujiyama. This is in Japan. It's called Mount Fujiyama Fitness Landscape, and it's rugged. You can call it Alps, Himalaya, whatever you like. Okay, but the point is, I demonstrated this only for 2 locus, but basically you can, if you start to construct this thing on higher dimensions, there are lots of local peaks, and most fitness landscapes are rugged. Okay, now I want to finish off going back. Okay, so let me just mention this. So there is all this data coming from experiments. You can also model these things, and there are models which we would like, which interpolates between these 2 limiting cases, in which you have a single fitness peak, and the ones which lots and lots of peaks. There's several models. I'll just give you reference. I don't have time for this, but one of them is called NK Fitness Landscape. Models for fitness landscapes. These are one of them, the most famous one perhaps, NK Fitness Landscape, which was inspired from spin glasses. But there are others. So this is Kaufman. Forget the name here. And there are others. Block model. And there are several others. Okay, so let me now come back to the equation for X sigma, because I want to sort of demonstrate one point. So I haven't put this mutation selection together. Before I do the equation, let's just see what to expect. So selection, as we said yesterday, if I had a high fitness, then there was this equilibrium between which one of the allele was absent, right? Selection usually has a tendency. So what does selection mean? It means if I have somewhere high fitness, population would tend to be concentrated over there. Is that your intuition too? Right? Because it's a peak. And again, from physics, it's like energy minimum, right? Okay. But mutation, what is it saying? It doesn't care whether you're good or bad. It just says, I don't know, I don't see a fitness. I'm just going to make 0 a 1 and 1 a 0. So if the population is sitting at the peak, mutation can spread it. It delocalizes it. So what do you have here? Again, from a physics background. Selection has a tendency to put things together, localize. Mutation has opposite competing tendency. What can happen? This is all of us. Huh? Phase transition, yeah. So let's see if there's a phase transition. Let me give you one example. Let's go back to this equation. So I want to demonstrate a phase transition in a steady state using this equation. And I'll just take one example, but many more you can do. So this is the example in which for a particular sequence, let's say 0, 0, 0, you can choose whatever you like. The value is W0. And for all the other ones, it's 1, and W0 is large than 1. Can you visualize this fitness landscape? There's just one sequence for which there's a high fitness. Everybody else has the same fitness, all the other sequences. So it's an extreme case of an epistasis. So I wrote something called alpha. When alpha goes to 0, you can check that you will get this landscape. It's called sharp peak landscape. Okay. Okay, so fitness landscape is simple, but that still doesn't mean that I can exactly solve this. And I want to do it in steady state, so I'm going to drop all this time. But there is one biological input which is useful to make analytical progress with this model. And that input is the following. If you have a wonderful car, which is very high mileage and everything, and then you just blindfold yourself or some friend of yours and give him spanner and hammer and stuff, and just make mutations in my engine. Would it increase the mileage? There's a small chance it might, but it's highly unlikely, right? Okay? Mutations are like that. At least I like to think of them like that. Most probably the mileage would decrease. Here, in biological context, it means most mutations are deleterious. This is something, again, which is borne out by experiments. That is, if I have a mutation, so I'm thinking of this fitness landscape. So everybody is down below, and there's just one sequence which has a high fitness. So mutations from this guy, the top guy, to the bottom ones, which are more likely, the top to bottom or bottom to top? Bottom to top would mean beneficial because fitness is increasing. Those are unlikely, right? What can happen is that this one, most mutations would be such that the guy at the top loses, okay? So that's the kind of thing that you could put in here, in your mutation matrix that simplifies things, and also work in the scaling limit. Mu goes to 0, L goes to infinity, so that L mu is finite. That this is true is also seen in experiments. If you measure per base pair mutation rate and multiply with genome length, at least for prokaryotes, that means the ones which don't have nucleus, this seems pretty good approximation. And there's one nice experiment where this value is seen to be 1 by 300, 330, something like that. I don't know. There's no understanding of that. If anybody has idea, I'll be happy to talk to you. Okay. But this is sort of, again, experimentally seen. But theoretically also, you can justify it. Let me just implement these two assumptions over there, and let's see what we get. So let me write down the equation for this guy. So this contains mutations from all the guys towards it, including itself. But I said I'm going to neglect the mutations which give the contribution to it. That means sigma prime is becoming sigma. The bottom guys are giving it to it. It's beneficial. We are not going to take that into account because of this. So I'm going to write this as Does this make sense? This is the term corresponding to Hamming distance 0, right? Mu to the power d, 1 minus mu to the power minus d, d equal to 0. But the mutations from a sequence which has one mutation, it has low fitness, that I'm not allowing because of biological reasons. And also it makes things analytical practical. So those terms is what I have drawn. In the denominator, I've used the fact that all the sequences except one have the same fitness. So the ones which has the highest fitness, that term is W sigma 0, X sigma 0. The rest have fitness 1, but because summation X is 1, that helps me to write the rest of the contribution in terms of X sigma 0. Is this okay? This is good because we can solve it. There are two solutions. There, this is so because it's common. Or when is this solution valid? I mean, there's a negative sign sitting here, so I have to be careful. W0 is larger than 1, but this could become negative. So if I say, because being a fraction, it should be positive. If I demand that this is a valid solution, when this is greater than or equal to 0, I find that this requires that Lu should be larger than log of W0. Sorry. So if the mutation probability, so again imagine, if mu was very, very high, so then you're taking away the stuff from the top guy, right? So it's just scattered around. It's just all over the Hamming space. So that's like, as if it's nothing, it doesn't hold a special position, right? It's like treating W naught as a special one. It's more like a flat fitness landscape. Everybody is safe. There's no selection, right? But if the mutation probabilities are small, then selection is able to keep the population at the fitness peak. And that critical value works out within that approximation to be this. So this is a sort of famous transition, and I forget the year. It's by this, is a Nobel laureate, Eigen, I think 1970, I'm not sure, I forget that. But again, you can find it. It's called error threshold transition. That means you need, that you lose the concept of a species, because it's all over the space. All genotypes, all genotypes are equally likely to be present. You lose this concept of species then, right? So that happens at a high enough mutation probability. Okay. So there are other examples. This is the simplest one. You could try with a fitness landscape which has just single peak. I'll just give you the answer. In that case, it turns out there's no phase transition. And I'll just write down the equation, and I urge you to try it, because it's a very standard model. Open any book, Opgen book, you will see a solution, and it's worth doing. It would help you to understand some of the things later. So I'll just write down the model, and please try. So in this scaling limit, if I had a fitness of this kind, I'll just write down the model for you, what equation you would get. For this fitness scheme, if you plug it in here, and I'm claiming this, it's possible to show that under these approximations, you obtain this model. Okay? And the reason I'm saying this is because this model is exactly solvable, both steady state and dynamics. And the answer is also rather simple. So I would urge you to try this whenever you have some time. Okay, I think maybe I'll just stop here because it'll just take me into a more longer discussion. So if you have any questions, comments, or whatever, you can ask. Okay. Diploid case, you have many stable equilibria. There is, I think there is a phase transition there also, of this kind. But if you like diploid case, there is an analog of this kind of fitness landscape for diploids, and there you can make some analytical progress. But it's not, again, the equations you get are very nonlinear. You have to put it in a computer to understand it. For diploid case, you can basically make progress with 2 loci. 3 and all, I have not seen analytical answers for that. That's extremely hard. Yeah, people have claimed it, but there are ifs, and it's not very clear that it's really so, because this mu-L, it means that mu-L is a constant, right? But experiments, more recent experiments show that it's valid, it's true only for certain class of organisms. It's not very generally true. So if you want, you can look at this person's work, Lynch. He has a lot of papers and some nice review articles. Yeah. sorry, I forgot the word. It's called quasi-species. I think, again, there's a concept in chemistry. So Manfred Eigen, he was a chemist. And in chemistry, there's a concept of species like water, I think, is a chemical species, in which things are very coherent, like they are very coherent things. But here, things are coherent, but they're sort of more cloudy in the sense things are not just sitting at the fitness peak. Everybody is not at the fitness peak. There are things at fitness peak and below, around it. So it's a more cloudy, hazy thing. So that's why the word, I think, quasi, it's not a real species like a chemical species. Like water is always H2O, right? It's like a delta function. But this is more hazy thing. So I think that's how this word called quasi came. So quasi-species would be when mutation probabilities are low enough.